Hello, everyone, and welcome to today's webinar hosted by the International Irradiation Association, or IIA. My name is Arthur Dumba, and I'm the IIA General Manager. I'll be your host today. We're happy to have presenters and attendees from around the world, and we appreciate you taking the time to be here. Before we begin, a quick introduction to the IIA. The International Irradiation Association is a not-for-profit organization that supports global irradiation industry across all technologies, applications, and sectors. We promote the safe and beneficial use of irradiation and serve as a bridge between science and industry, communicating, educating, and advocating on behalf of our members. Membership provides access to publications and resources, working groups, visibility opportunities, valuable networking opportunities, and discounted registration fees for IIA events such as IMRAP 2027, which will be held between the 12th and 16th of April in Dusseldorf, Germany. To learn more and get involved, visit iiaglobal.com or contact us at info@iiaglobal.com. Today's webinar is titled Radiation Microbiology in Pharmaceutical Products, and joined by Christine Walton, Nelson Laboratories Senior Scientist, Marta Winters, Nelson Laboratories Director of Science, and Chris Hardman, Nelson Laboratories Department Scientist. In this webinar, we will explore the purposes behind the various test methods and approaches, the testing associated with contamination control strategy, and how to properly combine the ISO and USP EP test method in the life cycle of a pharmaceutical product. A few housekeeping notes before we begin. This webinar is being recorded. A link to the recording will be shared with all participants shortly after the webinar is over. You can submit questions at any time using the Q&A function at the top of this screen. We'll have a dedicated session for question and answers after the webinar. We will endeavor to answer all your questions, but if we run out of time, we will circulate unanswered questions at a later date. The chat function has been turned off for this webinar, so please make sure to direct questions to the Q&A tab. I would now like to hand over to the Nelson Laboratories team.
Okay, well, thank you, Christine. Well, good morning, good afternoon, everybody. We're excited to discuss this topic. As a contract laboratory, this is these are questions and topics that we are regularly discussing with our customers, and I imagine others of you have probably had similar issues with this. So we're excited to cover this topic. We go, just to give an idea what we're going to cover here, and Arthur already went through this, the main topics we want to cover here. So the idea is we're going to focus first on some of the core concepts. So we're not going to talk specifically about radiation for the first little while we hit some of these really important concepts on test methods, sterilization, etc., and then we'll get more specific into the radiation sterilization side of things. So if we look at the primary focus, just to kind of set the foundation here, when we're looking at ISO versus any of the pharmacopoeia, and I don't know all the countries that are represented here, but when we talk about pharmacopoeia, we're largely use USP EP just to cover the bulk of those that are likely on here, but I'm sure there are some of you who are on who might have your own country's pharmacopoeia, and it might read differently than what we're used to here with the USP and EP. But just over the decades of time where we have the ISO approach to testing and sterilization, largely on the testing side, and then the pharmacopoeial side when it comes to testing, these two groups have evolved very separately. It's not been a collaborative situation with the evolution of how they tend to be used or how regulatory bodies tend to want to use them. So if we look at, for example, the USP EP concepts and where they work best. So this is not necessarily when they're being asked by regulatory bodies or when you might be using them, and this is where we feel this is where they work best. So aseptically processed products, and notice I'm not saying drugs because at this point you're probably all aware that it's more than just drug products that are aseptically processed. So if we just consider that we want our minds to be in the concept of aseptically processed products, where USP 71, for example, the sterility test is a prime example of a USP chapter that plays properly into this situation. But then we also have non-sterile products where there are some microbiological controls required, and the concerns are with both the numbers and in looking for objectionable microorganisms. We're going to come back to that. And generally, they need to be water soluble. So example here is like USP 61 and 62. So the focus, like I said, should not be on it's a drug, so we follow USP EP. So if we look at the ISO, when ISO works best for the similar chapters in microbiology. So for terminally sterilized products, the ISO works very well. I'm not sure about the EP. I know the USP has some chapters regarding sterilization, and largely they just point to ISO standards for details on how to comply regarding terminal sterilization. Now, ISO also has a chapter or a standard for non-sterile products with microbiological controls. So you see there's some overlap there with what it says up above, and for the ISO front, the concern there is with numbers and types of microorganisms. So you notice the main difference here is the USP EP, when we're talking about the general term bioburden, they're looking at numbers and are there objectionable microorganisms present, as opposed to ISO, which is what are the numbers and generally what types are present. And the ISO standard is set up to test both water soluble and non-water soluble devices or products. So an example there is the 11737-1. So again, the focus is not on it's a device, so we follow ISO. The focus should be on it's a terminally sterilized product, therefore largely we should follow ISO, but you'll see some overlap here. So on this first slide here, this is kind of a call to action. This is not a new thing, but the more we can start integrating people and approaches between the pharmacopoeia people and the device people, and of course combination products bring both of those together. But the more interaction we can have as we're working on these standards and chapters, the better we can overcome the confusion. Okay, so now I'll turn the time over to Christine.
Great. Hi, thanks, Martel. So as Martel was saying, before we get into radiation specifically, we'll kind of go over some of these general confusion points that are occurring. So as Martel hit on, there's some fairly clear pathways already set up. So we know for our pharmaceutical product what potentially the label claim is. That helps kind of direct into which category or testing modality we're going to look at. Terminal sterilization being supported by sterility assurance, and then that leads us down a path into potentially USP or ISO testing. Similar for aseptic processing, hey, we're looking at microbial control, and then another pathway for non-sterile products, with the focus being more on the microbial quality testing. That again tends to lead into those USP chapters. So different products are going to require different microbiological evidence, and then the different standards tend to answer those questions. So that's what we're going to kind of get into here, is how each of these standards have a different intent and answer different questions, and how we can capitalize on that to build an appropriate risk-based process and testing pathway, not just, hey, this is non-sterile, this is sterile. So that looked really clear, right? That cascading pathway, but confusion does happen very early in this process. So one of the biggest sources of confusion is that we really want to tend to label things really simply as sterile or non-sterile, right? That seems very straightforward. I know if something's sterile at the end, I know if something is non-sterile. The issue is everything before sterilization is non-sterile, right? So that's going to be non-sterile materials that are intended to be non-sterile as final products will obviously be non-sterile, but so is everything pre-sterilization. So that could be raw materials, excipients, any components prior to that sterilization product, excuse me, that sterilization step. So that can be a confusing step, is that non-sterile is inherent in the sterility pathway. So I would offer that we don't necessarily start with the question of, is it sterile or non-sterile? Instead, look at asking, what microbiological information are we looking for in a life cycle? So when we talk about life cycle, different stages of the product. So again, that upstream from the sterilization step, different life cycle stages will equal different microbiological questions we need to be answering, which will then in turn lead to a slightly different testing methodology. So that's, again, that clear cascading pathway maybe isn't so clear because the life cycle of your material will inhabit different areas as it goes through its process. So we know about different life cycles where it can be, but let's go back to that question for sterile products and non-sterile, and what the intent of some of those questions or what we're trying to answer with those tests could mean. So for sterile products, generally, again, generalized, but for these materials, for the sterilization process, we're trying to just simply show that product, that process is capable of producing the sterile product. And then for non-steriles, we're trying to look for population and patient safety and product quality. So, but they're going to follow, while the intent and the objective of these are quite different, they share intermediate steps. So again, that's where some of that confusion can come into play, that while the sterilization path might include a bioburden prior to sterilization, that's going to look very similar in its potential results and recovery as an enumeration step in a non-sterile pathway. But again, the intent of those end tests are going to be different. So those are going to, the bioburden might support the process validation, as opposed to maybe non-steriles are going to ask for specified microorganisms. We'll get into a little later, some not unheard of to have regulatory bodies actually ask for specified microorganisms on a sterile pathway, and some of the risks that can fall in there. And then the end testing of a sterility test versus non-sterile products, which are really just trying to show that they're meeting an acceptance criteria. So again, intent can be quite different, but that doesn't mean they're not sharing components through the process, which again can lead to that overall kind of confusion between pathways. So let's really, these are very generalized, but again, getting into the little bit of intent on some of these standards and methodologies that we kind of listed really quickly. So USP 61 is going to be that microbial qualification, or the quality. Generally tends to be geared towards the non-sterile pharmaceutical drug products. But that doesn't mean that's the only thing it can be geared towards, but that is the intent. So we need to understand how that intent feeds into the process. Same with USP 60, slightly newer chapter, and for Burkholderia cepacia complex, and USP 62. That's where you're going to look into, again, geared a little bit more towards those dosage drug products, but that's going to be looking for specified microorganisms. And then all of those falling under kind of the acceptance criteria guidance of USP 1111. That's where, based on the route of admission, it's going to outline potential pathways for determining acceptance criteria, which again, if you remember, for non-steriles is really kind of the intent. At what point is something safe and not going to be a danger for the patient or a danger for the material itself and changing how it functions. So, and then we have USP 71, which is going to demonstrate the sterility of not just aseptic processed products, but potentially others as well. Again, very general. And then we have the ISO's 11137-2. That's again going to be the sterilization validation, but has components to support other aspects. Same with 11737-1 and -2. Again, in its title, it gets geared towards healthcare products, but that doesn't mean it can't have components that can be utilized or intermediate steps that can be utilized for other, for pharmaceuticals or other products along this pathway. I did want to throw in some new chapters that were recently added just last year with 1119 and 1119.1. These were USP, bless them, we're trying really hard to help, but they acknowledge that there is definitely some confusion between bioburden methodology testing and then specifically USP 61. The intent of these chapters were to help clarify some of that confusion. I will leave it up to you if you think that was successful or not. I think what it did do is it allowed some generalization for building that risk-based analysis. It really opens up the pathway for, and really I think promotes what we're talking about here, which is going through your process specifically, your material specifically, and making sure that the path and the testing you put in place doesn't just follow the preset pathway, but builds a risk-based approach for your material. All right, so we've looked through all of the standards. We know that we need to be asking some very specific questions to make sure that we have an appropriate risk-based analysis of our material. So let's go through all of those questions that we need to make sure we're asking ourselves for our product. First of all, what is the material? Are we at a very beginning? Are we at raw material? Are we at an API? Are we looking at excipients? Are we looking at a final finished product? That will feed into the next, which is where is it at in its life cycle? Each of these will have different components in its life cycle, even obviously the finished product, but even an API or an excipient. Knowing where it's at will again help us for the next, which is which microbiological questions should we be asking here? So do we just care about enumeration? Do I just need to know what my levels are at? Is that a bioburden test? To help us start building our sterilization validation project. Is this non-sterile? So this is actually going to help us understand our acceptance criteria if we're within appropriate levels. Are we trying to find out or answer if we have a specific microorganism present, or are we really trying to prove sterility? Now that question will change depending on where that product is at in its life cycle, right? Understanding which of these questions we're trying to answer changes with the life cycle of the product. And then even more, sometimes a step I think we unintentionally skip over is what decision we'll be making from that result. It's not just the answer we get, but what are we going to do with it? So make sure that those are appropriately tied together. Am I going to use this enumeration to release? Am I using it to show that my process is in control? Or is it that bioburden result that's helping support a sterilization validation question? Are we using this for trending, which can actually be applied across the board back into a sterilization, back into control? Make sure that not just the question is being answered, but that we're appropriately assigning the right decisions to whichever result comes out of those. And then as we went through those standards, make sure that you're looking, that we're finding which standard answers the question, and it might be a combination. So I think we want to make sure we're pointing out that USP and EP and ISO are not competing standards. While they were not done in combination, and they do speak to very different intents, and the challenge can be knowing where to apply these in the process. It doesn't mean that we can't harness these microbiological questions that are being approached differently in each of these methodologies to make sure that we are creating the best path possible for the material and our specific product in mind. Just like in microbiology, it's always yes, but. So the but here is know the risks of using different methods for the same material. So we'll get into that in just a little bit. What I see quite often is that we've talked about life cycle. Hey, the regulatory body told me that I needed to perform some non-sterile testing. That makes sense because perhaps my product is in the portion of its life cycle where it's not in its sterilization step. So I need to make sure I'm showing control or I'm showing appropriate enumeration. However, so we know, hey, from that original slide a while ago, I know the non-sterile pathway, it's really clear, which means generally what happens is let's just modify USP 61 and 62. So I'd like to point out that risk and what the risks are with just moving forward with that process. So USP 6162, well-established non-sterile testing pathway. While it is geared a little bit more towards dosage drugs, you can make it fit other things. There is a little bit of room, but we need to make sure we are very well versed on the risks and where that can cause problems. So one of the biggest ones is actually the sampling plans. So the sampling plans for USP are designed very differently because they are designed generally for those water-soluble materials. They are designed for dosage drugs. They have very specific guidance on how you can control that with lot size, with dose. However, if you have a product that doesn't fit into that just exactly, maybe you'll look at ISO. But those, again, have different sampling plans based on the intent of those tests. So because the intent of those are to help support sterilization validation, those sampling plans will be designed very differently. And again, these in-process materials tend to not fit any of the approaches exactly. So the risk we need to look at is that applying one method outside of its intent or its purpose creates inconsistencies, specifically with those sampling sizes. Hey, 10 grams of a product as prescribed for in USP 61 probably won't read the same as it will for an ISO, sampling 10 samples versus a USP 71 that's asking based on different modalities and how it's being used. So if it's going to impact the sampling, that's obviously has the potential of impacting the interpretation as well. So make sure you are aware of all of these and how they can potentially impact sharing these test methods for the same product. Great, so we went through all of this. We said, please combine them, use good science, but then there's these risks we need to be aware of. So let's highlight what we can share between these methodologies and what we need to be a little bit more vigilant on. So what can be shared? Those microbiological techniques, the organism recovery principles, suitability, identification. We're in an awesome realm right now where we're getting into rapid methodologies, which are really opening up the world for microbiology, moving away from CFU into different unit qualifications. These are techniques that are well established, some up and coming with rapid methodologies, but that doesn't mean that these techniques can't be shared. So these methodologies, whether I'm following an ISO pathway or a USP or an EP, these pathways or these principles should be able to be shared and straightforward between them. The components that would be difficult or we need to make sure that we're not seeing them as simply interchangeable are going to be those sampling plans. We need to make sure that, yes, while I maybe followed a non-sterile testing pathway before, that methodology and that sample size, those quantities that I'm using, if I used a different acceptance criteria, that it speaks to my overall testing plan instead of creating an isolated result, which again, I can't make any decisions off of because it doesn't fit with my overall testing plan. Make sure we are understanding the intended use and how those quantities and those testing plans can actually imprint onto the intent of the test I'm using. And then ultimately that interpretation. It can't be a one-to-one, or if it is, we will need to make some different modifications outside of just trying to make it fit into an existing pathway. So to kind of call back to Martel's call to action, I would do a similar one here is let's use the science that's already there. We have well-established microbiology techniques, microorganism recoveries. We know extraction efficiencies. If say we're using a combined product that has a drug component as well as a medical device, that can muddy this water even more, right? Because now we're getting down into medical device pathways based or potentially more drug, even though a device is present. Use what is there to leverage it, but knowing the risks and how to address those. So a question we get all the time and what we see quite a bit is, hey, I have a regulator that asked me to follow a specific pathway. So I have a terminally sterilized material that they want me to run a specific specified microorganism recovery for E. coli. That might be totally valid because that's something we want to make sure isn't present in our material. So know when to push back And when to push back should be some of those components that we discussed that are not interchangeable. Understand how that modification would need to take place. Some of those modifications would have to be understanding the testing plan, understanding how it would need to move, as well as the quality, the quantification, how we would perhaps use an extraction efficiency on a modality for non-sterile that doesn't generally apply that. Doesn't mean that we can't. It means we need to use the science, use what's in place, and know how to modify. So I'm going to hand it over to Chris. Yeah. So kind of did a general background in what was there, some of the different pathways we have. Now we're going to get a little more specific into contamination control. Or I'm sorry, is this Martel? I apologize. That's right. Chris.
Okay. So let's look now into, like Christine mentioned, just a little bit on the contamination control strategy, or CCS. Again, just to reiterate that our focus is not on, is my product a drug or device, but that our focus is on what am I trying to accomplish, and what do I have around me that I can implement to help with that. So when we're talking about an overall contamination control strategy, this is a concept that is largely driven in the drug industry, and it's a great approach to whether it's like we've been talking about, whether it's a non-sterile product or a terminally sterilized or an aseptically processed product. They all will benefit from having an overall contamination control strategy, or CCS. When we look at this list of what we have there, these are all things that any type of product, usually we have these things in mind. I won't go through the whole list, but these are pretty typical things that at some level are present in processes. But what we want to accomplish here, if possible, is what we really need is that these different things speak to each other. This diagram here is from PDA Technical Report 90, and the concept here is that a contamination control strategy should be what we say holistic approach, meaning that we don't just have a disjointed list of things that we're looking at that are of concern microbiologically, but that we have each of them in place intentionally to meet a specific need, and that it's part of an overall program, not an isolated topic. So what you see here in the diagram, we won't spend a lot of time on this, but as scientists, we like that the very foundation of this all is scientific knowledge, and that's what we're trying to get at here. Make sure that your scientific knowledge is sound and is broad and deep, so that you understand how to implement things correctly. But as you work your way up, we see the contamination control layer there. That goes back to the previous list we were showing, the personnel, hygiene, process, product, design, equipment, etc. And as we're validating those controls and those processes, and then as we're monitoring those, we have specific criteria in place that might lead back to additional risk assessment in the contamination control and that contamination control band in this house that we're building here. So just want to get that concept in mind as we finish up these core concepts of what do we have, what are we trying to accomplish, and what do we have at our disposal to use to help gather what we're going to accomplish, or what we need to achieve. And now I'll turn the time over to Chris.
All right, thank you, Martel. So we've talked a little bit so far about kind of defining which path we're going to follow based off of the characteristics of the product or the process. And I'm going to talk a little bit about radiation sterilization. So kind of the cornerstone of our presentation here. So we've given you all the foundation, and now we're going to talk about kind of the first sterilization modality. And radiation sterilization is quite popular with pharmaceuticals. And it can be used on or finished products, or it could also be used on components that are going into aseptic reprocess samples as well. So kind of talk a little bit about this. So radiation sterilization is popular with pharmaceuticals because it does not react chemically with APIs or excipients. There are no residues, and there's less degradation that occurs to APIs or excipients compared to heat-based methods, which is another preferred method. But with some of the products or raw materials that are heat sensitive, steam or dry heat just isn't possible. Products can be sterilized in final packaging configurations if you're looking for a terminal sterilized product, finished good, but it can also be used, as I mentioned earlier, in process. And we do see it come through where we're looking at bulk APIs that are being sterilized through radiation sterilization. It also is great for complex drug products and delivery systems. So we have talked a little bit about it, but the combination products where there might be a med device component infused with a drug product, radiation sterilization can help for those non-water soluble devices. So I wanted to talk kind of high level to a radiation sterilization validation. It's also dose audit testing that's required thereafter. It follows a very similar testing path. So where we begin in the process is we perform bioburden testing to get a number of microorganisms that are present on those products. That follows ISO 11737-1. There are some supplemental tests that go into that as well, so we can look at potentially recovery efficiency depending on the product that's being tested. Christine talked a little bit about that as well. From there, we can use that bioburden data to set a verification to dose following ISO 11137-2. So we had that list of standards earlier. Hopefully this will kind of help to see where these come into play. After our verification dosing is applied to those products that we are trying to establish a sterility assurance level on, we move forward with test of sterility testing following ISO 11737-2 and BNF testing, which is pulled from 11737-2 as well, but is largely based off of USP 71. From there, if the test method acceptance criteria are met in 11137-2, we've established a minimum sterilization dose. And once this process has been demonstrated or that dose has been established, there is no need for sterility testing at the completion or demonstration that the minimum sterilization dose is acceptable for that product or component. So some regulatory challenges that we run into. So there's a void that currently exists in standards. So Christine touched on a lot of these during her portion of her presentation. So there's different requirements whether you're following USP or ISO. So there's differences in test batch sizes or those sampling plans. There's differences in the amounts of products that are tested. So generally with ISO, when we're following ISO, we're more concerned about what that sterile label claim is. So that can be the entire product, it could be just the fluid pathway, whereas a lot of USP guidance may be 10 milliliters or 10 grams, especially with those water-soluble products. ISO is also generally more concerned with microorganisms' radiation resistance, and that rarely correlates to pathogenicity of those same bugs. So it's pretty rare that an organism that's going to pose a challenge when trying to establish a minimum sterilization dose is also an organism that's of great concern or a high potential for infection in a patient. Another regulatory risk that or I should say gap that was identified that is currently being addressed in the new revision of the ISO 11137-2 standard that's currently in draft at the ISO level is when trying to establish a minimum sterilization dose, on bulk product, there was not a lot of guidance on what that should look like. So there is some new guidance going into that document to help help with establishing that minimum sterilization dose without having to do so on the entire bulk. So kind of bringing that focus back down to kind of a final dosage that you would expect to see in the final product. Next slide. So there are some challenges. I'm a radiation guy. It is very hard for me to admit, but there are some shortcomings sometimes. Very rarely, but I'd like to talk about those. So with radiation sterilization, electron beam, and specifically irradiating pharmaceuticals, electron beam can pose issues in being able to deliver an effective dose. So that could be the result of shadowing or complexity of the product, but there's a solution. Gamma may be an alternative for that product. So a lot of that will come into the material compatibility, the product, the API excipient compatibility of radiation in general. But there are options within radiation, electron beam versus gamma, that can be looked at to determine if there's one that's more suitable for your product. Radiation may degrade molecules of APIs or excipients. There is the potential to look at lower doses for the product. So if you have a fairly clean product or raw material, trying to drive that dose as low as possible to mitigate those harmful effects to the API or excipient. Or mitigation activities, there's also options at irradiating at lower temperatures. So that could be, on dry ice to help kind of mitigate those adverse effects as well. So packaging components or delivery systems damaged by radiation, a lot of times this can be polymers or different materials. Looking for alternative materials or additives to those specific components that are affected is a good solution to kind of allow for radiation to be a more viable process for those packaging components as well. Next slide. So there are options. I hope the takeaway here is, radiation is a great method and it can be used. Sometimes there is some troubleshooting that needs to go into that for any of those products where it's deemed that a sterility assurance level needs to be claimed or sterility assurance level needs to be claimed, or a sterile label claim made for those products or components going into that manufacturing process, even for aseptically processed samples.
Okay, thank you, Martel, Christine. Okay, so some quick question. Does ISO 11737 reference any aspects of the USP stroke EP?
Let's see. I know we don't normatively. So when you look at the normative references in the -1, and Arthur, was the question specific to 11737-1, or was it also -2?
Both, yes.
Both.
Generally.
Okay. Yeah. So in the bioburden standard, there is no strict reference there. There might be a reference when we're talking, like in the bibliography, there might be a reference because sometimes there are inhibitory substances, and the pharmacopoeia sometimes have good details on overcoming inhibition in a test method. But it would be a kind of an indirect reference in part 1 for bioburden. But part 2 currently does specifically reference the pharmacopoeia because, like Chris mentioned, that there's such a good approach to determining the method suitability, or what used to be called the bacteriostasis fungostasis or BF test. But that has led to some confusion. So in the new version, so we've not started yet, but we plan on starting a revision to the ISO 11737-2 probably next year, is because we're wrapping up the revision to the bioburden standard. Hope to be finishing that up this year or early next year. So close on the heels of that, we're going to start revising part 2, and we do intend to bring over all the concepts from the USP or the EP into the ISO, just because of the confusion that's arisen. Is the pharmacopoeia talk about using 2 different temperatures and 2 different media types, and at the ISO level we're often just using 1 temperature and 1 media type, and we see too much confusion happening there when they're saying when we reference the pharmacopoeia. So it'll look the same as far as the microorganisms that we use. At least that's the direction we are likely going. Same bugs, same approach, but specifically it will be the ISO temperature and media type. Chris, I know you're involved with some of this as well. Anything to add to that?
No, I think you covered everything that I would have hit on. So yeah.
Yeah, as the USP girl at the table, I would want to point out that USP is actually fairly good at referencing ISO. I would also point out that there's kind of a treasure trove within USP, within the monographs that are present, particularly when you're looking at your raw materials. And it has a really strong chemical background as well. So while those monographs tend to not reference ISO specifically, they are going to give you some really good guidance on what specifically those materials have been established to look for. So that might help with some of that crossover as well.
Okay, perfect. And does the USP mention SALs 10 to the -6, or is that specific to medical devices?
Yeah, that's a good question. If you look at USP 71, and then 71 is harmonized with the EP, and I think there might be other countries where that chapter on sterility testing is harmonized. There is no reference anywhere in that chapter that passing the sterility test results in a sterility assurance level. And if we go back to the intent behind the pharmacopoeial sterility test, the intent the focus should not be on, I have sterilized a product, terminally sterilized a product, now I'm using USP 71 to try and prove sterility. There's a huge gap there. If you're testing 20 or 40 samples per the USP sterility test method, trying to prove a 10 to the -6 SAL, you can't achieve that. And that's why that's useless data to perform a sterility test on fully sterilized product, because a test of 20 or 40 samples, you've demonstrated an SAL of, and I don't want to run the math here really quickly, but it's probably like 10 to the -1.3 or 1.4, something like that. So you're proving 10 to the -1 point something, but trying to prove 10 to the -6. And how do you bridge that gap? And so the USP approach, the pharmacopoeial approach, has never been to prove an SAL. It has been to meet the requirement to perform a test for sterility after an aseptic process. And those of you that know aseptic processing, there is never a sterility assurance level or a PNSU, a probability of non-sterile unit, associated with an aseptic process. You either meet the criteria for passing or don't. There is not a number associated with that. So it's a long answer to that, but I'm glad that question was asked, because it's a common point of confusion that, for example, an aseptic process is an SAL 10 to the -3 because I've tested 1,000 products as part of my process simulation, and that's just not the case. Scientifically, that's not a valid approach.
That's also where that interpretation becomes confusing when you're interchanging these, right? So suddenly I think I'm allowed to make an SAL claim simply because of numbers, when because of the intent of that test, it really doesn't cross between them.
Excellent.
Okay. And the last question, what's the main difference between test for sterility and test of sterility?
Chris, you want to tackle that one?
I'll do my best to. Yeah, so test for sterility, kind of what Martella touched on, right? It's typically, test for sterility is typically associated with aseptically processed samples. There is no allowable limit for positive tests for sterility. It's everything has to be negative. Whereas when we're approaching or taking an SAL approach, similar to, we'll just say radiation sterilization, for example, and we're targeting a specific sterility assurance level, there's probabilities that are incorporated into that sterilization process and establishing those SALs. So that said, when we're doing a test of sterility for a radiation sterilized product, there's acceptance criteria with allowable positive tests of sterility that we can still show that we're demonstrating at least that SAL, if not a lower SAL, making sure I'm using that terminology correctly. So for instance, a lot of times for like VD max, we're targeting an SAL 10 to the -1. So you'll test 10 samples for a test of sterility after exposure to a verification dose, kind of that I had displayed in that infographic. So a sublethal dose, if you will. With that, you're allowed 1 positive out of 10 samples or 2 out of 20. So those SALs do allow for some positives, and the test of sterility does allow for positives because it is a probability approach, a mathematical probability approach, whereas a test for sterility is you have to demonstrate that all samples are negative for that test of sterility based off of the batch size that's defined for that test.
So it's probably geared a little more towards a process as opposed to final result.
Correct.
We love to apply statistics, but it doesn't work everywhere. So understanding, again, that risk and applying that math inappropriately to the result.
Okay, thank you all very much. That's all we have time for today. A special thank you to our presenters for taking the time to be with us and provide their insight in today's topic. And thank you to everyone watching for participating in today's webinar. The recording for the webinar will be circulated shortly, as will the follow-up to any QA sessions. Contact information for myself and our presenters is shown on the screen, and the webinar will officially end in a few minutes. So you have time to note down any contact information you may need. Thank you again for joining us, and we look forward to seeing you at our next webinar. Goodbye.
Thanks, everyone.
Thank you.